Hello all, welcome to Department of Parasitology, Faculty of Medicine. Today our class is on tissue protozoa, Toxoplasma gondii, which causes toxoplasmosis. I'm Lau here, and if you have any questions later, you can contact me at this email address, lauyeeling@um.edu.my. We start our class now. Right, here is the lecture outline for today. Everything will start with introduction, followed by epidemiology, morphology, pathology, clinical manifestation, and then diagnosis, treatment and management, and also the prevention and control. Toxoplasma gondii is a single cell parasite, causes toxoplasmosis. It is perhaps the best known for its connection to cats. This parasite can move from its feline host to humans and other warm-blooded animals. According to Wong and Rimanton in 1994, more than 90% of the world population are infected by this parasite. It causes flu-like symptoms in mild cases, and most of the time it is asymptomatic with no clinical symptoms. This disease has more severe consequences, such as congenital birth defects, eye disease, or potentially fatal in congenital infection and immunocompromised individuals. A form of disease called toxoplasmic encephalitis. It is primarily due to the reinfection of the latent toxoplasma infection in immunocompromised individuals. Alright, let us look at the life cycle of Toxoplasma gondii. We start with the definitive host, which is the cats. In the cats, cats are usually infected by tissue cysts as well from other infected tissue of other animals. So from there, the cats the parasite will form oocysts by sexual cycles in the cats because it is a definitive host, and these oocysts will be shedded into the cat feces 3 to 34 days after the cat became infected. The oocysts will only be infective after 2 to 3 days after it's shed into the soil or water. Okay. The intermediate hosts, such as human and other animals, birds or rodents, can become infected after ingestion of this parasite through the soil, contaminated soil, water, or sometimes plant material contaminated with the oocysts. From there, from the soil, the parasite will transform into the tegizoid form once enter the intermediate host. In the tegizoid form is the active form where the parasite can multiply and disseminate throughout the blood or lymphatic system to the other tissue in the body. And eventually, it will settle down in the tissue of the intermediate host and form tissue cysts, which is the dormant stage, the pedizoid stage in this case. Usually, it will be deposited into tissues such as muscle, myocardia, or CNS. Okay, cats is the definitive host. Usually, it happens in domestic or wild cats that roaming around. We can pick up the infection from infected rodents mostly, and it will shed the oocysts in the feces, as I mentioned just now. And humans, we are the intermediate host, which usually the asexual cycles of the parasite life cycle happen in human. There are two forms in humans, which is the tegizoid, the active form, and also the pedizoid, the dormant form. This is a little bit fun fact about Toxoplasma gondii. In rat infected with this parasite, has shown that this kind of rat is usually lost their typical fear of cats, and especially their fear for cat urine. So research has shown that the parasite can cause the rats to be less fearful of the cat or the cat urines, and because of that, they are even being attracted to the smell of the cat and they go closer to the cats. This may be a way of how the parasite can manipulate the intermediate host so that they can be eaten by the definitive host, which is the cats here. It is funny, right? So this is just some facts about research being done on Toxoplasma gondii. Let's continue with the life cycles. As I mentioned just now, the tetrazoid form, this is how it looks like under the microscope. So the tetrazoid forms is usually crescent or oval in shape. It is 3 times 6 micron meter, and it is rapidly dividing and invading other cells. So it causes the acute infections or the active infection. This state can be destroyed actually by freezing and thawing. So just freeze store the meat that have the tetrazoid, it can actually destroy the tetrazoid itself. So the predizoid form is the tissue cyst form, which vary in size, depends on how many predizoids or how many several thousand of predizoid in one of these tissue cysts. So it is sometimes dormant, and even though it's dormant, the cell is the tetrazoid, the zoid is still slowly dividing, and it's usually in brain cells, heart, or muscle cells. So the host carrying these states or parasite is actually having a chronic or latent infections. And the oocyst is the one that in the feces, cat feces. It contains many sporozoites, and it is usually oval in shape. It's very hard to find these oocysts in soil. Even by us, we look through so many soils. This form is actually very hard to obtain. We can see sometimes in the water as well. This form can be easily destroyed as well by boiling or burning or using some ammonium solutions incubated for 3 hours. Okay, here is what I'm going to repeat about the life cycles. Oocyst is the form that shaded from the cat feces, and it can be transmitted to cat litter, soils, or water when intermediate who ingested it by contaminations of hands, garden vegetables, or water sources. And these oocysts will become tetrazoids in the intermediate host and actively spread in the blood system, and from there this tetrazoid can transmit through ingestion or infected milk or by blood transfusion or transplantation and also transplacenta. And tetrazoid form will become tissue cysts in the less active forms eventually. And these tissue cysts can be hidden in body tissue such as muscle, kidney, heart, and lungs. And once the host ingested the uncooked meat or raw meat, it can ingest this form of the cyst. It can also be transmitted to transplantations, where the infected organs is donated to the recipient. So what are the risk factors contributing to transmission? Of course, contact with the cats, consuming uncooked meat, blood transfusion, organ plant transplantation, and congenital infection is the way of transmission. The risk group involve pregnancy, immunosuppressed patient, prolonged steroid, chemotherapy, and those that having chronic diseases, HIV, AIDS, and transplant recipients, they are all the risk groups. So now we look at the epidemiology of toxoplasmosis. And okay, because a lot of the infection is no symptom, there's asymptomatic cases. So the reports on how many cases of toxoplasma gondii infection worldwide is actually very hard to know. And many cases go unreported. So a study done in 2002 is shown that about 40% of the world population are infected, and eventually it go up to 90%. Okay, in this slide here is two studies done by our previous lecturer in our department, Dr. Virano Nisampaton, and it showed that in Malaysia we have about 30% of population are infected with this parasite, and in Thailand about 20% are infected. So if you look at the map again, 90% is very high. 90% of Europeans are infected with this disease. Of course, it's mainly due to the consumption of uncooked meat, the raw or uncooked beef, or they like to eat steak. So especially in countries such as France, the infection is quite high, right? And then in North America, similarly, they have a culture of eating raw or uncooked or half-cooked meat. There are about 30 to 60% of toxoplasmosis in these countries. Here is the pathogenesis. Toxoplasma gondii oocysts are ingested in material contaminated by the feces from infected cats. Oocysts will then transform into tegizoid shortly after the injections. Tegizoid multiply asexually within the host cells by repeated binary fission or endodiogeny, leading to pseudocysts. Host cells will rupture when it could no longer support the growth of the tegizoid and invade other cells. Tegizoids are disseminated throughout the body tissue via the blood and limbs to other organs such as skeleton, muscles, myocardium, brain, and eyes. Eventually, petezoids are formed in these tissues. Most cysts occur in the muscle and brain, can persist for many years. This is the chronic phase of the infections. Once in a while, the cyst will rupture and release when the host immunity is weak. If humans or other carnivores eat meat containing cysts with petezoids, then this stage emerge and invade cells, and they divide as tegizoid first, leading to the acute phase and then to the chronic phase. In congenital toxoplasmosis, during acute infection with tegizoid in the bloodstream during pregnancy, the tegizoid will colonize in the placenta and cross the barrier to reach the fetus in 30% of the cases, leading to congenital toxoplasmosis. Okay, now we look at the clinical manifestations. In congenital infection, this disease causes maternal parasitemia and parasititis. In the first trimester of the pregnancy, it causes abortion, which is quite severe. So if it happened during the third trimester, it causes congenital toxoplasmosis. And the baby may be born with hydrocephalus, cerebral calcification, retinal keratitis, cataract, and each other. This is a baby. It's a picture of a baby showing the hydrocephalus symptoms. And immunocompetent or sometimes is in ocular patient, they are usually asymptomatic. It causes non-specific illness. Sometimes it will cause lymphadenopathy, myalgic encephalomyelitis, and ocular diseases, and some very mild diseases. Okay, most of the time the disease causes heavy problem or severe problem in immunocompromised patients such as HIV and AIDS patient, which causes focal cerebral and brainstem, hemioparesis, dysphasia, or movement disorders. Sometimes in the severe case, it may cause cerebral dysfunction, lethargy, confusion, coma, and cognitive impairment. Right, it will also cause sometimes it will also cause neuropsychiatric disease, causes anxiety and as well as psychosis. Okay, one of the toxoplasma most frightening or most controversial effect is impact on the mind. Okay, if a woman is infected with a parasite while she is pregnant, her fetus may be at risk of developing schizophrenia or other mental disorder, according to a study done in 26 on the people living in Denmark, and it was published in the Journal of Biological Psychiatry. And other study also show that in 2014, which is published in the Journal of Nervous and Mental Disease, it found that toxoplasma antibodies are very high in patients with schizophrenia disease, and it provides the evidence that there's potential link between the parasite and mental illness. This is very interesting, right? Now we look at toxoplasmosis and HIV patient. It is a forever a living legacy between these two infections. So toxoplasmosis is the most common opportunistic parasitic pathogen that causes the focal brain lesion in HIV patient. Most of the cases are reactivation of the latent toxoplasma infection. It causes life-threatening disease which causes to death if this misdiagnosed or no diagnosis at all. So how to diagnose toxoplasmosis? We can do it by isolation of the parasite and then inoculate into the animal host, let it propagate, or we can also culture the parasites and look at the histological examination, looking at the serological test. And now we have polymerase chain reaction, which is highly sensitive, which can detect the infection in many types of samples, blood samples, CSF samples, aqueous humor or urine samples. And this disease can be diagnosed by looking at CT scan, MRI, and PET scans as well. So there are many serological tests available, including ELISA test, agglutination assay, direct agglutination assay, fluorescent antibody test, or indirect agglutination assay, so many types, heme agglutination assay, and also ISAGA. So when you look at the antibody titles, it peaks at different time. Of course, IgM come early during the first to fourth week of infection, followed by IgG after 2 to 3 months of infection. And it is very sometimes confusing because IgG can prolong for a long time in the body, sometimes could be years. So in this slide here is showing IgG can be detected even after 6 years of infections. There are many ELISA kits available to detect different types of toxoplasmosis. In this table showing ocular toxoplasmosis can be detected by serological test IgG, IgM. Congenital toxoplasmosis same as well. There are many tests, and of course immunosuppressed patient also there are many ways to test the so diagnosis in pregnant women is very important because the clinical manifestation is different when you get it early during the early infection of the pregnancy, or if you get it in the late stage of the pregnancy, the manifestation may be different. So because of the result, the uncertainty of the ELISA, IgG, and IgM test, we have to further test the patient with another test, which is IgG avidity test. IgG avidity test is measuring the strength of the binding between the antibody and the antigen in the test. The stronger the binding is indicating that the IgG is already present in the body for a longer time. So a high IgG avidity result can actually mean that the pregnant woman is already having the infection at least 12 to 16 weeks. So this can actually back up with your IgG and IgM results saying that this infection is already caught sometimes. Okay, a low IgG avidity result sometimes can indicate a recent infection because the binding is not that strong yet, but it could be interpreted wrongly as well. So just be careful when you use also ELISA test or IgG test, IgG avidity test, because the IgG avidity can also prolong for many months after infections. Now we look at the diagnosis in HIV patient. Most of the time, the diagnosis in HIV patient is empirical, which means we cannot truly confirm the infections. So we have to look at many angles. So you have to first look at the clinical presentation of whether there is any neurological deficiency, and look at the anti-HIV antibody, whether the CD4 is less than 200 cells, and look at the CT or MRI scans, look for the ring lesion, and also depends on whether the patient is responsive to anti-toxoplasma therapy or not. Or of course, you back up with also of serological evidence, IgG, IgM, or IgG, IgM, or both. So brain biopsy is reserved for patients who fail to respond to specific therapies. It's rarely done. Okay, it will only be done when it is needed. It really needed. For treatment of toxoplasmosis in congenital disease, uh, the babies or the newborn can be given sulfadiazine or pyrimethamine or Fancida with folic acids for 1 year. And for treatment in pregnancy, during the first trimester, we can treat the pregnant woman with pyrimethamine. In the later trimester, treat with sulfadiazine and pyrimethamine, followed by folic acid as well. And optionally can be given pyrimethamine until terms. In immunocompetent individual, usually is asymptomatic, but if the patient is presented with symptoms, it could be treated with sulfadiazine and pyrimethamine as well. And in ocular patient can receive the same treatment as well for a month. Similarly, with treatment on HIV or AIDS patients, for acute therapy, we use the same thing, pyrimethamine and sulfadiazine, and back up with antibiotic, clindamycin. In Malaysia, we use clindamycin and Fancida. And the patient can be continue with primary prophylaxis, chlorotrimazole, and secondary prophylaxis for maintenance, pyrimethamine and sulfadiazine as well, or Tyson chlorotrimazole after the X or T treatments. So this is the last slide which showing the prevention and control. This is especially important for pregnant lady or woman planning for pregnancy. Always eat cooked food, or if you still prefer raw meat, you can actually freeze and thaw the meat many times in the freezer. Avoid drinking untreated water. Wear gloves during gardening or in contact with soil or sand, okay, because it may be contaminated with cat feces. Keep all the outdoor sandboxes covered. Feed the cats only with canned food or dry, commercial or well-cooked food. Okay, advise a pregnant lady or woman looking forward to become pregnant to screen for Toxoplasma gondii. All right, that's the end of today's classes. Thank you very much.